Hi, I wanted to introduce Bernie Fisher because he hasn't publicly been introduced today, and I wanted to say that he's the lead medical officer in the Division of Psychiatry Products in Cedar. I'm very happy to be co-chairing this session with him today, and he's going to start out with taking you through the questions. Thank you, Bernie.
Okay, good morning. So it struck me listening to some of the comments that have been made about yesterday and about some of the limitations. I had a professor in medical school who said, with regard to psychiatric diagnoses, as soon as you have an idea about the pathology, it's not a psychiatric diagnosis anymore. General paresis is now infectious diseases, mixed edema psychosis is now endocrinology, Alzheimer's disease is generally treated by neurologists. And so without some concrete idea of the pathophysiology of some of our psychiatric disorders, it's really hard to do things like fluid biomarkers and PET imaging. So as exciting as yesterday was, as a psychiatrist, I'm even more excited about today because we're going to talk about things where I think we're a little bit further ahead than we are in fluid biomarkers, EEGs, and we're going to talk a little bit about clinical biomarkers as well in the afternoon. So just to go through the questions quickly, things to think about during the presentations. What's the state of the science for these EEG measures? Are these ready for prime time? Are we ready to roll these out and start doing clinical trials with using EEGs? What are the challenges for quantifying and qualifying these EEG biomarkers? Then, how do we improve the business case for doing these EEG biomarkers? So for industry, thinking about investing in this equipment, in this technology. And then finally, what are the similarities and differences between qualifying EEG and neuroimaging, and can then be used maybe in tandem to produce a multimodal biomarker?
Thank you very much. This has been a wonderful day and a half so far. Thank you for inviting me to speak, and look forward to the comments as we go along. Disclosures. So let me just start by couching some of the issues we've been wrestling with in the field of psychiatry. And it's interesting to see, so I'm a psychiatrist and a neuroscientist, so kind of live on both sides of this. And interesting to see how both sides deal with the core issues that we have in psychiatric therapeutics, unlike, in some ways, what you heard about from Parkinson's and Alzheimer's. So we don't have a DAT marker that tells us about progression of the disease. We're not even sure if the definition of our disease is correct, and if progression has anything to do with treatment. So let me contextualize some of this for you. In 2008, the field got thrown into a lot of turmoil in the world of depression when Irving Kirsch published a meta-analysis that looked at all the data that was submitted to FDA. And his conclusion, as captured by The Guardian here, is that antidepressants don't work. The effect size was around 0.3 between drug and placebo. And the conclusion is that they're really not much, antidepressants are really not much better than placebo. So a lot of heartache ensued. And then Andreas Cipriani did another meta-analysis almost 10 years to the day since the first one. The conclusion was completely different. The conclusion here is that they do work, and if you read in the bottom there, it says, it's official, antidepressants are not snake oil or a conspiracy. So just saying what it is about the way we communicate with each other over those 10 years, the more interesting fact here is not the differences in conclusion per se, but that they're actually the same data. The effect size between drug and placebo was still 0.3. Our conclusion, out of perhaps more desperation and the state of the field evolving, changed. So let's rethink this question a little bit, put it in different terms. Maybe the question isn't whether an antidepressant works, maybe that's not the right thing to be thinking about, but whether depression works. Whether the idea of giving a drug to a depressed person necessarily makes sense by how we are defining the illness. So I'm going to argue that that's perhaps where our attention is better placed, for the moment at least, and just start by giving you context. So I'm going to present, I changed the title a bit from the placeholder. I'm going to present some work on the study called EMBARK, but also other work But let me just first tell you about EMBARK. EMBARK is a multi-site NIMH-sponsored treatment prediction study in depression. Took about 300 people, randomized them to sertraline versus placebo, about as plain vanilla as you get. And what you get at the end is about as plain vanilla as of a result, which is the drug-placebo difference is roughly an effect size of 0.3 with a slight 10% or less improvement in response rate for drug versus placebo, exactly what you would expect. If you look around, today we're talking about EEG, if you look around at what we have in the literature with respect to the utility of EEG for predicting treatment outcome, there was a very nice recent meta-analysis from Alec Widge in AJP, which showed an ROC curve like this that suggests that there is something there, that EEG can predict treatment outcome. But in the end, that was actually an extraordinarily non-robust finding, as they highlighted is really due to small samples, inflating the effect size, very kind of inconsistent and poor method consistency, and also really no validation at all. So maybe there's something with EEG, but I think we need to prove it rather than assert it. If we're going to do EEG, and as Greg nicely, I think beautifully sort of laid out, EEG is an old technology, but as I'll try to argue today, EEG can come into the 21st century from the 1970s by virtue of machine learning. But we have to think about how we go about machine learning with EEG with an idea of what it has to offer. And part of what it has to, thinking about what it has to offer is how to get past the core challenges of EEG. Number one is this issue of volume conduction. So there's a smearing of the signal at the brain because you're detecting scalp signal from multiple places in the brain at the same time, and there's not a one-to-one relationship. That's not necessarily a game stopper. With every form of imaging, and probably a lot of fluid biomarkers, is a tremendous need for dimensionality reduction. That is, you have many, many, many things that you could measure at the sensor level, the frequency level, what have you, and you never have that many samples. So you have to think about dimension reduction in an intelligent way. That also will affect how well you can generalize. And then from a machine learning perspective, there's the issue of optimization. So how do you get to an answer that is globally optimal? How do you get there consistently and get the same thing every time? And that is non-trivial. You do not want to be stuck in what are called local minima in life and in general. So this is how EEG machine learning is normally done. And I'll show you a novel approach we created for these data. So normally, you might take EEG, extract features, in this case, let's say, the power at a given electrode, fit it to a prediction model and test it on cross-validated data. The problem here, number one, is that it's a divide and conquer strategy. You determine features on the one hand, and you fit the model on the other, and these things don't connect with each other. Number two is you've put into this the idea that channels are the level at which the feature is to be found. That's already an assumption. You square the amplitude to get power, and now you've made a nonlinear problem out of an assumption. That is, I think when they say when you make an assumption, you make and then you can fill in the blank of what you make out of you and me. But assumptions in this case are particularly problematic because we need to discover more out of the data themselves because they're very rich. And when you assume some of the structure, you do constrain the solution. And importantly, the traditional approach in dealing with EEG really does not leverage the spatiotemporal structure of EEG. Structure in terms of locations in the brain, frequencies, timing, and covariance, and so forth. So what we developed was a new machine learning model, taking a cue actually off of brain-machine interface literature, that uses something called spatial filtering. I'll illustrate what that means in a second. And we tried to solve the various challenges I presented to you, volume conduction optimization and dimension reduction, as one process. So mathematically, this model can actually be proved. That is, my postdoc really went through literally mathematical proofs to be able to get a single end-to-end optimizable convex solvable process. In other words, there is a global minimum that you can achieve consistently every time in a computationally efficient manner by dealing with spatial filters, which I'll show you an example of in a second. You can deal with volume conduction, considering basically the whole montage at the same time. And the way he did dimension reduction is something called supervised dimension reduction, and I'll illustrate that in contrast to unsupervised methods. But the idea is to find the way to reduce the dimensions that best fit the prediction goal as opposed to the best fit the data. So we end up fitting our models with about 10 different signals, more or less, instead of hundreds of thousands or millions of signals. And as I'll show you, it makes a big difference. So this is just to give you a sense for spatial filtering. What you see there are two brain sources. The one on the left is the signal. The one on the right is noise, just as a toy example. And you're recording signals on the scalp. A spatial filter considers the whole montage at the same time, looking for what's signal and noise, and figures out a weighting for these signals in order to recover that true signal that we're interested in. So we're no longer working at the single channel level, we're looking at the covariance between all the channels. In some ways, you can actually relate the model that we created here to a single hidden layer in a deep learning network, except we've encoded information about the spatiotemporal structure of the EEG. All right, so that's a little bit about the machine learning. Let me show you some results and kind of go through this and then another approach as well. So we took the folks in the sertraline arm in EMBARK, did a 10-fold cross-validation, train on 9/10, apply on the 1/10 that were left out. And I'm showing here the correlation between the true change in depression scores and the model predicted change in depression scores. As you can see, they're highly significant results, 10 to the minus 12th, with a strong predictive relationship. But now you can start having fun with this, because it's not just about finding the signal, and it's great. This is the biggest study of its kind with EEG that we know of, at least in the public sphere. And the machine learning model does work and predict outcome with sertraline. But you want to know a lot of other things about this model. For one, you want to know what happens if you take this solution and apply it to the arm where people got placebo. Do you similarly predict outcome with placebo, or is the outcome drug specific? The other thing, and I'll show you that result in a second, the other thing is you want to show that this is better than just what we do normally, which is measure severity. And so we've done that in a variety of ways. We've shown machine learning applied to symptoms, even at the item level, does not predict outcome anywhere near as well. But also this prediction, this EEG signal that we have in the model, is independent of severity of depression at baseline. We find this over and over and over in our studies, that what predicts outcome is not necessarily what differs between a patient and a control. All right, so you take the model, apply it now to the placebo arm, and you see that there is zero prediction of placebo outcome. In other words, this EEG machine learning signal is specific for predicting sertraline outcome relative to placebo. We also could take advantage of the fact that EMBARK was a 4-site study across the United States. And let's call this a feature rather than a bug. Each of the sites used a different EEG system. Don't ask. It's what happened. But it's an interesting opportunity. So now you can take 3 sites to determine the model and test on the 4th site, arguably a worst case scenario, because you would never do a study with every single site having a different device in an actual pharma study, right? And it turns out it still works and is a highly robust model. Slightly weaker, but this is really a worst case scenario. And it's also a nice test of generalization, because these are people from completely different parts of the country on different equipment. So let me just summarize this for you in the same kind of graph I showed before. So this is all comers, 42 versus 34% response rate or so. If I take the model, the cross-validated model predicted outcome, and I separate into quartiles of the model predicted outcome, and then look at the real outcome in the cross-validated subjects, what you find is as follows. Here I'm splitting by lowest quartile, middle 2 quartiles, and highest quartile, basically showing the distribution. And you can see the folks in the highest quartile of predicted response, they have a response rate of 70%, double that of placebo. They should be the ones getting sertraline. The ones in the bottom quartile should definitely not be getting sertraline, right? So this gets at the issue of heterogeneity in depression, but as, and this will need to be replicated in future samples and so forth. But as we think about this and think about stratifying our clinical trials based on these kinds of signals, one would argue that the drug does work. It just works for this biologically defined group of people, independent of their disease severity. Now, medications aren't the only treatment we have For depression, we have brain stimulation, something called repetitive transcranial magnetic stimulation, and people use different protocols for this. So how do we link what I've just shown you to a different way to intervene with the brain in depression? Well, one intermediary, one way to try to understand what that resting EEG signature even means is to relate it to how the brain responds to perturbation, not with a therapeutic RTMS, but single TMS pulses delivered to different parts of the brain while recording simultaneous EEG. Just to give you a heuristic, this is what happens when you stimulate the prefrontal cortex. The video you're going to see is 100 milliseconds of brain time source localized to the brain in terms of the cascade of EEG events that happen when you stimulate a single site in the brain. This allows us to measure the causal influence of stimulating that brain region with respect to all of these downstream events. So now, what if we look at that brain response and see if it relates to our resting biomarker signature? We noted when you look at these latent signals, these spatial patterns that we use to fit the model, that on the left there is the most positively weighted spatial pattern, and on the right is the most negatively weighted spatial patterns. One thing that should actually pop out is that the signal in the back of the brain is both positively and negatively weighted. So that's an interesting kind of example of finding noise to cancel out. But one of the things that was unique to the negatively weighted signal is a prefrontal heavy, lateral prefrontal heavy component, which happens to also be where you might do therapeutic stimulation. So we looked in a separate sample of depressed patients who got TMS EEG, concurrent TMS EEG probes of different brain regions, shown here, 4 in the prefrontal cortex plus motor and visual cortex, and saw whether the strength of their resting EEG treatment prediction pattern with this big prefrontal component correlated with the response to stimulation in their brain in different regions. And in fact, it did, but only for the prefrontal stimulation sites, arguing that this resting signature may be telling us something about the function of the prefrontal cortex that's relevant for treatment. So we found another archival data set from a colleague, Martijn Arns in the Netherlands, which had EEG at baseline, in this case with 26 channels instead of the 54 channels that we had in EMBARK. And we could ask what the relationship then is of the medication signal to outcome, not with medication, but with RTMS. That is, if you apply the sertraline signal to these people who did not get sertraline, they got RTMS, 1 Hertz RTMS in this case, what is their outcome with treatment stratified on the medication signal, knowing also that in prior work of ours and others has been the suggestion that the things that predict brain-wise response to antidepressants go almost in the opposite direction in predicting outcome with RTMS in small samples. So we're stratifying in exactly the same way as I'm showing here, but now the outcome with RTMS, especially for anxiety symptoms, is opposite. The people who are going to respond well to sertraline respond the worst to RTMS. The people who respond the worst to sertraline, or predicted to respond worst to sertraline, are predicted to respond best to RTMS. So that's interesting. It could lead to treatment selection, but certainly help us understand the mechanisms of treatment as we develop novel ones. A few important methodological points. Channel count matters. So this is taking the EMBARK data and just taking subsets of channels, the full 54, 32, or 21 equally placed channels. And what you can see is that the prediction drops off. So if one were to design a study prospectively, I would argue for going more like 64 channels than really low channel count, despite the temptation to reduce time and costs and so forth. I think it pays off in the end data-wise. The other thing is that not all machine learning and not all signals are created the same. When we looked at different ways that people in the past have determined EEG signals that are put into machine learning, for example, using band power, like alpha power, at the electrode level, which is what I was saying one should not do just on principle reasons. Also, for practical reasons, that does not predict treatment outcome with sertraline. Andy Luchter's measure, which is a variant of power called cordance, also at the electrode level did not predict treatment outcome. But, so these are the sort of simple ways that one can find signals. Those don't work when you subject them to the rigors of machine learning. But also, the issue of data reduction it matters a lot how you reduce data. We did a supervised data reduction. If you do unsupervised methods like ICA and PCA, you don't necessarily find the signals that predict treatment outcome. In fact, here, there's really no significant prediction. All right. So I'm going to sort of transition a little bit here to applying this kind of machine learning method in another context, back to that RTMS example here, and see if we can actually think about this as a way to, as a sort of COU, to determine which of multiple protocols a person getting RTMS should get as, say, a new way to go about doing RTMS, not to exclude people, but to say, well, you know, I could do the right of the brain, the right side of the brain, the left side of the brain, doesn't matter to me. I just want to know which one is better for that person. So in this archival data set, 26 channels, we had data on 1 Hertz RTMS on the right, which is what I showed you before. If you fit a model to predict that directly as opposed to through sertraline, you get a very nice prediction that actually outperforms what you would expect from the EMBARC data based on the number of channels. We also had people who got left 10 Hertz RTMS, and there again you can predict treatment outcome at about the level you would expect from the channel number. These predictions are both independent of severity of depression at baseline or the use of concurrent medications at baseline. And I can speak to that part later if you're interested. So let's do an experiment with these data, because in a potential outcomes framework, you can get the predicted outcome with either of these interventions on cross-validated samples, even if you did not actually get that intervention. So we have an opportunity to do an in silico reassignment of people based on their machine learning result, finding that if we do that, that about half of the people should have gotten the protocol that they weren't assigned to. In other words, clinicians were basically performing a chance in assigning protocols, sorry. If you had gotten 1 Hertz but should have gotten 10 Hertz or the other way around, you saw a tremendous increase in your response rate, consistent with a cross-validated prediction. And the result of that is a 10% absolute rate of response increase, or number needed to treat of 10, using an optimized versus a non-optimized treatment, in this case still all active treatment, there's no sham here, all in silico, in a study with a high response rate at baseline. So this is promising for how you would use biomarkers to select between treatments that otherwise would be in equipoise using the same kind of machine. I don't know if this is like question time or if I have a couple minutes. It's because it's, okay, good, because I have more data. All right, now we heard about fMRI, and I grew up in part as an fMRI person, but we're particularly interested in trying to get into the clinic, doing discovery in the clinic, translating rapidly into the clinic, and fMRI, as wonderful as it is, presents challenges. For the past few years, we've been working on methods to try to get past that, to use EEG to tell us about things that we already have a deep understanding of from the nice work you've seen, for example, from Ty and from Alan on fMRI connectivity. And got very excited with this finding. This is a method in MEG that creates connectivity networks that look like and correlate with fMRI networks by looking at the power envelope, that is the instantaneous amplitude of an oscillation. So that little line above the oscillation is its instantaneous amplitude. You use that as your seed for a given region and look across the brain to see what's connected to that, using a very clever trick that they created here to get past volume conduction, which is a tremendous, tremendous challenge in EEG and MEG, and they did it quite cleverly. To show you then what happens when you develop this and validate it for EEG, let me just give you a couple of examples. So this is what you see if you seed the visual cortex. You see bilateral visual cortex. You seed somatomotor cortex, somatosensory cortex, you get the bilateral sensory and motor cortices plus higher order motor areas. You can get the default mode network. You can get the frontoparietal executive network. So these are networks that we know and love from the fMRI world, but now we could do it on high density EEG acquired in clinics. To get kind of a first proof of concept of the utility of this approach, we looked at connectivity much as has been talked about on the fMRI side, looking at a variety of regions across the brain and how the whole connectome looks. but now in a group of about 100 PTSD positive veterans and 100 or so combat exposed veteran control, so really well-matched group, and just seeing whether as a whole PTSD differs from healthy people, knowing that there's heterogeneity, which I'm not going to deal with here. There's another interesting story there. But let's just see whether we can detect something using this tool and what does it reveal to us. So I'm going to look at carrier frequencies, the connectivity, power envelope connectivity using carrier frequencies ranging from theta to low gamma in the eyes open and eyes closed condition with a massive correction for multiple comparisons across this whole thing. And when you compare groups, what you find is that almost exclusively the group difference is in the theta eyes open condition. Let's seed one of those regions and see what it looks like. It literally paints the prefrontal cortex, almost to the exclusion of everything else. So here we're seeding the left prefrontal cortex, and you can see that in PTSD, their whole prefrontal cortex is underconnected, but only in theta eyes open connectivity. One of the things we understand about theta as a frontal theta in general with EEG is it's related to cognition and cognitive demand. And so most psychiatric disorders have a deficit in cognition, as do these subjects here in the digit span task. So we were able to then test whether individual differences in theta eyes open power envelope connectivity within these group difference regions also maps onto cognitive task performance, and these were the corrected results that did. In other words, a lot of this signal seems to be a mapping between frontal disconnectivity and cognitive dysfunction in these patients, again, not related to symptoms, related to objective metrics here. Finally, going back full circle to EMBARK, we can ask the question of what if we take this kind of connectivity and ask in EMBARK, do we find network patterns that tell us something about treatment prediction and treatment change? And the answer again here is yes. These are, everything here is corrected for multiple comparisons. So here are patterns, very parietal, interestingly, of connectivity, where the connectivity moderates between treatment outcome, that is, differentially predicts sertraline versus placebo outcome. But just the same, you could look at the change in EEG connectivity with treatment. In this case, in EMBARK, we had 1 week into treatment assessments, not post, unfortunately. But even at 1 week, you can tell that a different network is engaged by drug versus placebo. So these are differential changes in drug versus placebo. All right, just to kind of wrap it up. Hopefully I've argued a bit for why EEG is useful to consider as a biomarker. It's a rich source of information. It's much cheaper to implement. I think Raquel made this point yesterday. It can be done at point of care, even in remote places. Greg showed that very nicely. It's a direct measure of brain function, which I think has a lot of virtues as we think about designing, doing drug development around EEG. But to utilize EEG in the best way possible, you really do have to think about its spatiotemporal structure, and it's not just any old data that you throw into a machine learning algorithm, and really create EEG-tailored machine learning approaches, doing these new kind of connectivity methods, things that think ahead a little bit, and really focus on scale and reproducibility, because there are a lot of EEG data actually out there that can be gotten and harmonized. So I'm going to stop there. Thank all of the people who've been involved in this work, very generous funding sources as well, and just put out that if you're interested in using EEG for drug development, I would be more than happy to have the conversation with you. Please send me an email, full name @stanford.edu. Thank you very much for your attention.
So our next speaker is John Sweeney, who is professor of psychiatry at UT Southwest and adjunct professor at University of Cincinnati, and he's going to be telling us about EEG-based measures in psychosis spectrum and schizophrenia.
Hello, everyone. Thank you for staying for the second day, because the information overload from this meeting, I'm sure for all of us, is pretty intense because we're covering such a broad range of topics and perspectives. So hopefully everybody's electrodes are still green. Okay. I am going to continue discussion about EEG, and I have had a few advantages here because previous people have already covered things that I plan to talk about, which gives me a little bit more time to ramble on about things I would like to add. So First, let me tell you about a few things about myself and other things that I've done before I get to my data. I am across several institutions now at this later stage in my career. The first one that I note there is my involvement in the radiology department. So I also have a role of adjunct professor in the radiology department at West China Hospital, which is in Chengdu, China. And the reason I go there is that there's an opportunity to do research on scale that's really just not possible here any longer. So, for example, we are now gearing up to study 1,000 never-treated schizophrenia patients to look at drug effects and using MR as a biomarker to predict outcome. Now, for those of you doing studies here in the United States, we're down to about 5 to 8 never-treated patients that come in because of primary care takes care of giving the meds, and so we can't do these kind of studies at all anymore. So the chances to look at biomarkers in China now, where there's a centralized healthcare system. Chengdu is a nice city of 20 million people, nice small Chinese city. And with a relatively centralized healthcare system, so we can do studies there that aren't possible. So I think I wanted to mention something about those. I think for all of us planning our talks, we kind of said, well, we'll give our usual kind of academic talk, but I think a lot of you in the room who aren't embedded in psychiatric biomarker research might benefit from a little bit more of an overview of things that other people have found, which I like to say some things about in the time that I now have available from not having to go through other slides in such detail. So one of the things that we have done here is looking at heterogeneity. So a big part of our research program is tried to focus on resolving heterogeneity. We start with the assumption that our psychiatric symptoms are general broad classifications mostly from smart German people a few hundred years ago who basically describe people with problems of thinking and people with problems of unhappiness. And there's just massive biological heterogeneity. I think that's just clear to everyone. The genetics are highly heterogeneous. Every biomarker we looked at the presentation is highly heterogeneous. So there's heterogeneity in these syndromes that needs to be resolved. Now, as a nosological problem in psychiatry, that's kind of a straightforward thing you really need to do something about. But from a drug development perspective, this heterogeneity means there's heterogeneous biological substrates of disease. And if you start with the idea that you're going to have a drug to target a particular mechanism in a broad clinical syndrome where the biology is so heterogeneous that you have absolutely no idea what percentage of the population is likely to be affected by the target that you're trying to engage, you have a real limitation. So one of the, I think, really important strategies, and it's a bit different than Amit's, which was, by the way, absolutely great talk, is to try to use the biomarkers to resolve heterogeneity in the population and start at that point. So what are some things we've done? Well, one of the, and I can't talk about all this, of course, but one of the interesting things we've done is we have large samples of never treated patients in Chengdu. So we have over 100 PTSD patients, over 100 depressed patients. We have a couple hundred never treated schizophrenia patients. And in all three of those studies, when we've used unsupervised machine learning to look at the MR data, what's been evident is that there's subgroups of patients with increased, widely distributed cortical thickness, so increased thickening of the cortical mantle, not a reduction, and then another group of patients with reduced cortical thickening. So, those are just heterogeneous findings, qualitative subgroups formed in each of these populations. Now, we've also done a study like that in Cincinnati with our bipolar patients, and identified the same thing, a group with increased cortical thickness and a group with decreased cortical thickness. In that subgroup with increased cortical thickness, their response to antipsychotic medications was considerably better than those with a reduction. Now, we didn't find that with the lithium-treated patients. So with that specific subclassification, using the MR grouping of people based on cortical thickness patterns, we can identify a subgroup of people who respond effectively to treatment with antipsychotics. So there's promising findings coming out. There's another whole line of studies looking at MR to look at drug effects. So there's not only is there long-term beneficial effects of antipsychotics that we can see, especially in the dorsal attention system, but there's adverse effects on executive functions in some patients. That's consistent with when the similar things have been looked at in monkeys. It's related to negative symptom worsening in some patients. And to a degree that we found in candidate gene studies that glutamate genetics predict the subgroup of people that are going to show that adverse effect. So the ability to use our biomarkers not only to predict who's going to respond to treatment, but who's going to have an adverse response to treatment is really important information for clinical management and also for monitoring patients during trials for evaluating treatment effects. So there's a lot going on in the MR world. That's interesting. This is just another aside is looking at the relationship between the connectivity. Amit was mentioning the connectivity differences. You can look at connectivity with MR as you would with EEG. And in our EEG studies and our MR studies, looking at frontal temporal connectivity alterations is a very good predictor for whether people have recently had auditory hallucinations. So the mechanisms of auditory hallucinations are involving a particular circuitry. We can look at that, and we can look at the biology of a particular crucial symptom and a positive symptom in psychosis. So there's a lot going on here in smaller sample studies that really provides a good framework for moving on. So in our Cincinnati studies, I just mentioned quickly, we're studying neurodevelopmental disorders, and we focused on Fragile X. Fragile X syndrome is the most common single gene basis for autism and also for intellectual disability. And because that we know what the mechanism is, we can have a knockout gene model. So we can study electrophysiology from the organoid to the slice physiology level, to the in vivo physiology measure in the knockout mouse and in the human. And we can establish homologies, and then we can we're starting now to test drugs across that platform. So electrophysiology, we're learning things from that whole mechanism, that mechanistically about the approaches for that kind of thing. But there's really important promise for using electrophysiology in studying translational medicine approaches. So just to mention those things. But moving on with what will now be a shorter talk because of that time and because I don't need to cover a good number of my slides. There's been a large interest in biomarker development for schizophrenia. So this is straight out of PubMed. And you can see around the year 2000, we just had a rapid acceleration in people interested in biomarker development for schizophrenia. Now, we talked some yesterday when I had the microphone a bit at the end of the day, and others have talked about this, of course, a lot too, but we have real problems in biomarker development for psychiatry that we need to resolve and think through how to deal with and move forward. So first of all, the big one is that psychotic disorders are complex and biologically heterogeneous. And this is a big limitation. Also, the biomarkers we have are largely empirical. They're not, as in many of the neurology studies we heard about, where the biomarker is trying to target a known mechanism. We are trying to identify the mechanism. Ty mentioned that earlier. It's an important thing. The other thing is that a lot of our biomarkers are not specific. They run true across disorders. So we don't have a disorder specificity to most of our findings, really, if not all. And so this is the kind of circumstance that's led us to be really focused primarily in our work on trying to resolve the heterogeneity. So that's really been the big focus of our work. So I'm going to take you through one study where we tried to deal with this problem, and this is our BSNP consortium. We've been at this for a while. Multi-site study, and I'll try to take you through it quickly. So first, this is a slide that we all very much enjoy because we were 15 years younger when we took these pictures. So it's nice to show a picture of yourself like that. Notice I had brown hair. So a group of us got together and developed this. The people in the top row designed the initial study along with Guni Thaker, and we added Elliot Gershon and Jeff Bishop for the genetics, and Brett Clements and Sarah Keaty later. So the basic structure of our study was to say, well, if there's heterogeneity across disorders, can we use biomarkers to resort cases in an informative way, irrespective of what clinical psychotic disorder diagnosis they have in DSM? So So that was basically what we wanted to do. And so we recruited a good number of people. This is the first wave of studies. We had basically close to 1,000 patients. So we looked at 1,000 relatives to look at familiarity of these abnormalities and healthy controls. The second wave of recruitment is about 80% done. There's approximately, we will soon have more than 2,000 more participants in a case control study. Patients were relatively clinically stable, so it was limited acute illness effect. Pharmacology was stable for at least a month before we tested patients, and we excluded patients with things that would confound data interpretation. And data is available on NDAR, by the way, from the first wave of studies. We used a multidimensional kind of exploratory neuroscience approach to this study, collecting cognitive eye movement, a lot of eye movement studies in psychiatry that have really been interesting, EEG, MR, and DNA studies. So I'm going to obviously focus here on the EEG studies going forward. So as you can think from as I explained the rationale for the study, we basically were going to take psychotic patients with schizophrenia, schizoaffective, or psychotic bipolar diagnoses, pool them into a psychosis group, conduct machine learning studies, and see if we could identify subgroups of people. And this is a complex study. It went through 7 rounds of review at the American Journal, so it was kind of, there's a supplement online that's kind of like the Bible or something for everything we had to explain. But there were 3 subgroups formed. I want to focus your attention on the left 2. The one on the left sample was a group where their electrophysiology was diminished. So there's reduced activity. Present a tone, their ERP is reduced. Those people with reduced auditory ERPs were the same group that had alterations in cortical thickness and reduction in cortical thickness. So they're the group that has more structural MR abnormalities on T1 studies, and a specific profile of auditory activity abnormalities. The second group, in fact, had a complete opposite pattern. Instead of having low background activity and low ERP responses, which is basically a hypofunctional neurophysiologic system, the second group had increased ERPs and increased background noise. So we're using now in this large sample, which is, I think, really a real plus of all 3 of the talks today, looking at really large samples of patients. We're able to pull apart neurophysiologically distinct subgroups of people. And the treatment the relevance of this remains to be established. But if you think about excitatory inhibitory imbalance as a target for many drugs that we're developing, the idea that you have a subgroup of people who have excessive excitability and a group that have reduced excitability lets you know that it would be very helpful to know this information a priori to stratify patients before you begin a trial. So this will show you the data in quantitative way. If you focus on the right, which is really where the key findings are, the red is what we refer to as biotype 1 with a reduced responsivity, and you can see on the right for the basic auditory responses that they're markedly reduced selectively in a subgroup of people, and that in the blue in the resting activity measures, you see a marked increase, with 0 being healthy control values. So you can see that a lot of the times when we do these machine learning studies in psychiatry, we kind of pull out an unaffected, a mildly affected, and a more severely affected subgroup. This is really different. These are neurophysiologically, qualitatively distinct subgroups of people being pulled out of a very large sample. We did an analysis recently to show that we can replicate this classification pattern in a second sample of 1,000 people. So these are robust classification differences. So lessons learned, subgroup stratification using EEG is possible, which is really going to be crucial for stratification. As others have shown, the multi-site EEG studies are feasible with quality data. The DSM diagnoses, the syndromal diagnoses, can be misleading when large and meaningful subgroups of people exist within them, which really needs to be considered from drug development and for development of nosology and psychiatry. Okay. So I'm going to shift to one. Do you know how much time I have Okay, so I was asked to talk about the Prodrome work, and Ty is in the room, who really led the study to a large degree, I suppose. And so I called Dan Mathelon, who's a friend of many of us in the room, who actually did compose a lot of these analyses and papers. So I want to tell you about, summarize what those findings are, and thankfully he loaned me some slides to do that with best information currently available. So we used, I'm going to focus on the mismatch negativity, which was a key measure in their prodromal studies. And now I'm getting into areas where people have talked about this before. You've heard about the Prodrome study from Ty, and Greg actually presented his slide on the top right from showing the differences in also in a study of about 2,000 people. What the controls are looking like on the bottom. I suppose I don't have I'm tempted to. Well, anyway, on the bottom of the black line, you can see robust mismatch negativity effects for the healthy controls relative to the reduction in those effects above the line that are pretty robust and consistent across people, with a mismatch basically giving an occasional deviant auditory stimulus and leading to a significant response in healthy individuals that's significantly larger than in affected patients with schizophrenia, and also I'll show you in people in the Prodrome. So Dan Mathelon also, or not Dan Mathelon, Dan Javitt, working in this area, loaned me a few other slides to give you an idea of the effect sizes of this mismatch negativity effect. They're relatively high, consistent across studies, consistent across parametric manipulations. So in the order of 0.7, 0.8, not bad for an electrophysiological biomarker performance. And you can see here that there's a lot more people looking at mismatch negativity. I'm not sure they're quite exponential functions there, but for Dan, he thought that would be a good way to make the point. This slide was also presented yesterday to show that mismatch, Greg used this data to show that mismatch effects were robust in the patients, but also what their functional implication was. So the mismatch effect is the most robust measure of early sensory processing deficits that we have. And it's associated with cognitive impairment, and the cognitive impairment's associated with social functioning. So this is a clinically relevant sensory electrophysiology measure. So it's really not a geeky thing to measure in EEG labs and undergraduate departments. It actually is pretty related to clinical functioning of patients. The abnormality is very clinically relevant. The mismatch can readily be tested in rodents, so this can support translational medicine development using this approach. This is a study where a few electrodes were put over the relevant cortical area. But I can tell you that in our Fragile X program, we've shifted to looking at 30 lead EEG studies on a mouse, which is really pretty impressive. Couldn't quite do that when 20 years ago. So we're doing 30, it's Devin Binder's work in his lab, but there's 30 lead studies. And one of the interesting things is once you have more electrodes and you can average effects, you can, he's finding that his drug effect sensitivity with his mice is doubled. So he needs half the sample sizes to do his studies when he's looking at his 30 lead system. The other thing that's important is that, as Amit was talking about, looking at connectivity measures is really getting to be important, and is something we can do with the computational tools we have. Amit mentioned that it's really important. And our Fragile X work, for example, when we're just looking at the ERPs or resting power, we can pull probably about 40 to 50% of the patients away from healthy norms, from all healthy people. But when you use the connectivity measures, we're like up to 70, 80, 85% of our patients are completely outside the range of healthy people using the connectivity measures. And most of the things that we've looked at for EEG, for biomarker development, have been simple ERP measures. And these kind of connectivity tools that we can look at now with high density leads are potentially really going to be informative, as Amit showed with his depression data, and as is also the case in our neurodevelopmental disabilities program. There's also been a lot of pharmacology studies done looking at mismatch. Dan Javitt has really been the leader of this, and he's really looking at it as a measure of NMDA hypofunction. Basically reducing ketamine challenge, for example, you shut down you don't shut it down, you reduce the mismatch effect quite considerably, and there's a number of different drugs that he's looked at with that. So there's interesting preclinical evidence that the mismatch effect is a sensitive tool for looking at drug response, especially in the glutamate NMDA area. So one of the other things that's obviously important about our focus on biomarkers is in our use of the tools for clinical practice. So Dan was really interested as part of the Naples Consortium to look at the degree to which these EEG abnormalities would be useful for predicting conversion to psychosis. So the study has already been described of basically looking at people at risk for psychosis as Ty described for his Naples study. There's not a lot to say about that. I need to say about that other than it was kind of a heroic effort in a large multi-site consortium to identify a biomarker and clinical predictors of risk for conversion, which is obviously crucial importance. If we can stop people from converting to psychosis early on, this would be a huge advance for the field. So the mismatch effect, basically presenting an atypical, a different response like beep, 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 beep. An odd event at 500 millisecond interstimulus interval. And this is the summary of his effects. He used a specific effort to look at a double deviant approach where he varied tone duration and tone frequency simultaneously to get a bigger mismatch effect, which was helpful. But what you can see is that if you go down from the top row to the bottom, from the healthy individuals to the people who went on to convert to psychosis, that there was a significant, at time of recruitment, a significant reduction in mismatch effect in those who went on to convert to psychosis as a group that was different from what you see either in the at-risk individuals who recovered as Ty showed, or those who maintained their level of prodromal symptomatology without converting. So this was a promising effect. So if you look at the group level data, you find nice significant effects in p-values, and it publishes well. But when you get to the AUC data, the utility for separating converters from non-converters using this method was not very impressive. I mean, compared to other things, it was not the most successful thing in that regard. The data was quite reliable. So from the mismatch effect, that did not pan out as well as some of the MR data that we heard about earlier. So another paradigm is basically doing something similar to the oddball, but presenting it with longer ISIs. Longer time between stimuli. And what you can see here is effects that are a little bit different. And I spent a fair amount of time talking with Dan about this on the phone. But you can look at the top row and see that when you look at the converters, there's again, as there was at the group level in the mismatch data, there's a significant reduction in their P3 response in those who went on to convert to psychosis. But what was interesting here is that the group that really stood out as being very normal was those who remitted, those who recovered. And I think a lot of us are thinking about, well, we want to identify people who are going to convert to go into trial. But it also would be very helpful to be able to identify subgroups of people who are less likely to convert, who are not going to convert, and get them out of trials so that you can increase the percentage of people who are going to convert in your study and have a better chance of a drug response to reduce that. So So while the mismatch might be effective for identifying people with a problem is going to convert a little bit better, this approach, this paradigm seemed more useful in identifying people who were not going to convert. As you can see in the lines on the top right, the average ERP waveforms, that the non-converters and the controls are basically overlaid. So it's kind of a different potential use of this application in trials, but one that looks really pretty promising. Again, AUCs are not going to blow you out, and again, the ICCs at 1 year follow-up are pretty solid. So ICC is a relatively conservative reliability parameter. So it seems like this is like, so going over all this, all said and done, kind of where are we? So I think there's multiple promising advances from EEG studies of psychosis syndromes, both at people who have converted and who are ill, that we are able to identify biologically distinct subgroups of people that are of particular relevance for thinking about trials focusing on any drug that's likely to increase or decrease the cortical excitability. So basically, any drug going after a glutamatergic or GABAergic drug would be particularly interesting in that regard to have this stratification potential. And as I had time to start out earlier, the MR data for subgrouping people potentially informative ways for trials is come along pretty well also. So my kind of take about this is that after 50 years of discovery science, and we've been at this with imaging studies for about 50 years now, and electrophysiology even longer, we're at a point now where we can begin a shift to applications research, and it's starting now, as you hear from our talks today. That, 5-10 years ago was not at all true, but there's a lot more interest in trying to determine what's the practical utility of all these measures. And you sit and think about, well, where are we in broad brushstrokes? You think about Winston Churchill when America decided to enter World War II, and Winston said, well, this isn't the beginning of the end, but it is finally the end of the beginning. And I think to a large extent that's where we are here. We have a lot of promising leads. We have growing consortia, large consortia studies showing feasibility of multi-site studies. We have promising findings from these that are identifying biological parameters relevant for predicting treatment outcome. And I think we're at a point where we can start working together. And I think it's really valuable to me and impressive and gratifying that we're talking as a group of academics and FDA and industry representatives really thinking about grappling with this problem, that if we don't get these biomarkers into trials, we're likely to be struggling with the same problems of failed trials that we've had for a long time because of the heterogeneity of the disorder. So I think it's a good time to have this meeting, and with all the progress that's been made in smaller sample studies and some large consortium studies now. So future directions, the computational methods. I mean, we've gone from ERP measures to the kind of thing Amit was talking about, and we're doing in our studies as well. The computational tools with high density, high quality EEG recordings are really exceptional now, and we don't have to write C code to do it. We can use MATLAB, so it's feasible to actually make some pretty steady progress. And there's programming and shared programs across laboratory groups is really accelerating and increasing the quality of what we can measure and explore in data that we already have. So we have other issues. The testing in trials is really needed. Another consideration I wanted to raise is that from the perspective of using these biomarkers, if you think that schizophrenia, for example, is a heterogeneous illness, what you want is a high sensitivity, high specificity biomarker. If you think there's subgroups of people that are biologically distinct, you want a low sensitivity, high specificity biomarker. So if you're only looking for high sensitivity and high specificity biomarkers, you may be throwing out everything that's important in the data. And that's why thinking about whether this subgrouping idea is important and considering that in your use of biomarkers is really so important. So some of these standard mismatch approaches, resting state, P3 studies, I think those have been used for so long that from a technical perspective and implementation perspective, with some reasonable decisions about standardization, we could move forward there. I think getting these kind of measures into proof of concept studies collaboratively with industry is really important, so we can all together start getting a feel of their utility, because largely that's not really explored, but it really has potential to optimize and de-risk phase 3 trials. And I think lastly, as many people have said, all of these tools for acquiring data, whether it's MR or EEG, and for analyzing data, whether it's MR or EEG, are in a rapid state of evolution. And they're giving us new measures to exploit. but a lot of them, I think, are on the analysis side, especially in EEG. And with high-density lead studies, we can just evolve our analysis of the data without having to shift our technology so much going forward. So I think that's a fair assessment. I think so. So thank you.
So our next speaker is James McPartland. He is a professor at the Yale Study Center and also directs the Yale Disabilities Clinic there. He's also the principal investigator on the Autism Biomarkers clinical trial, which is a Biomarkers Consortium project in the Neuroscience Steering Committee. He's going to talk to us about EEG and autism.
Hi, thanks. It's really fun to be here. I think Diane observed, and I hadn't reflected until then, psychiatric disorders in this room are an unusual space. And so I'll talk to you about a pediatric psychiatric disorder, which is an especially unusual space. I was asked to talk at a kind of a big picture. So as context, this Autism Biomarkers Consortium that I direct, we will conclude this coming month data collection over the past 3 years. Autism, as I'll make the case, is a disorder I think that's behind a lot of the disorders that we've thought about, even psychiatric disorders in terms of understanding biomarkers, and yet we submitted in November a letter of intent to the FDA. So I think it's a good test case for thinking about what you can do with the information that you have. And this is what I want to talk about. A really quick primer in autism with a specific eye towards issues of relevance for biomarker development, the kinds of contexts of use that are desired in autism, what are some of the challenges specific to biomarker development in autism, and reflecting actually on the things we've heard today are really not unique to autism, but common to many psychiatric conditions, and then where things stand with EEG biomarkers in autism, and I'll talk about a particular biomarker called the N170. It's an event-related potential, and this is the one that we've put in a letter of intent, and then I'll talk about what I see as some potential next steps for biomarkers in autism. So autism spectrum disorder, it changed in 2013 with the DSM-5. So I study a disorder that exists as it is written in the textbook, right? So our biology is based on inference of a description of a set of behaviors. These are the behaviors really with this level of specificity. The deficits in social communication and social interaction, children have to show difficulties in 3 of 3 symptom clusters with multiple symptoms in each cluster. Restricted and repetitive behaviors or atypical sensory response. Children just have to show 2 symptoms in 2 areas of 4 areas. It has to be present in the early developmental period, again with that level of specificity. And these symptoms can be present at the time of an evaluation or any time in this person's history. So what a mess. Right? So lots and lots of different symptoms that we could look at. You don't have to show the same symptoms in 2 people to meet a diagnostic criteria of autism. You don't even have to show all the symptoms in a person at the same time. Right? So it's a really, really complicated disorder. And for that reason, as you'd expect, we have a tremendous degree of heterogeneity in what the clinical phenotype of autism looks like. In some ways that are unrelated to those diagnostic criteria. We have wide range in intellectual ability in autism, with many children with autism into intellectually disabled age, range many well above. In terms of language, there are children with autism who will never develop any spoken language, children with fluent language. There are co-occurring conditions that are very common in autism. Most adults with autism meet clinical levels of anxiety. Many meet depression. Schizophrenia can happen in autism. So I guess the point is that I could be up here giving a talk about biomarker development for any one of those conditions, and those are the things that just happen as co-occurring conditions in autism. And then even if we didn't think about all that stuff that's extraneous to the core diagnostic symptoms, the symptoms themselves can manifest in really different ways. You can have 2 people who meet criteria for autism, and one has no interest in other people, and the other is excited about approaching people, will do so in unusual ways. But really, so there is very little ground truth in autism. We try to think about what the biomarkers are measuring, what the disorder is. And the way that we try to quantify our ground truth is also inherently problematic. Really, the way that we measure symptomatology in autism, our gold standards are really a semi-structured interview, which is a fancy way of saying a play session with me. And I rate things like eye contact, I rate things like social ability. We can also do an interview with parents. These two, the ADOS, the Autism Diagnostic Observation Schedule, and the Autism Diagnostic Interview are really the gold standard for research characterization in alongside DSM-5 diagnosis. And so we use those. Those are, when we think about clinical trials, those are the entry into a clinical trial, getting a diagnosis based on those criteria. And then we really want to think about quantifying symptoms in a continuous way or measuring change. We either rely on these kind of play sessions again, or we use caregiver or self-report questionnaires. So really, we're not measuring a lot of biology in a biological disorder. What are the reasons, what are some of the goals for biomarker use in autism? And I've tried to map these onto the best terms, even though they may not fit perfectly. Well, one is susceptibility and risk. So when I look for all these behaviors in autism, I'm looking for behaviors that don't emerge in typical development until 18 to 24 months. So really, we're handcuffed to do a decent job trying to diagnose autism in children any younger than that. But presumably, there are neural precursors, or there could be other things like metabolic or genetic markers that we could find that would indicate earlier in the course of a person's life, or prenatally, whether a person is likely to develop autism or will develop autism. And that is our best predictor for intervention now, is the age of intervention. So that's a goal. That's a holy grail. Pharmacodynamic response is it's overkill to call it that, but I wanted to map it on to the best terms because there really are no pharmacotherapies for the core symptoms in autism, but we don't have good tools for measuring whether our treatments are behavioral treatments, and we still don't have good tools for measuring whether they work. So remember, any treatment, whether it's a drug, whether it's behavior, is treating the central nervous system. We presently don't quantify treatment response in the central nervous system. We measure it in behavior. We measure it using questionnaires that aren't designed for this purpose. What does it mean to report on change in a 6-week trial with a parent questionnaire that asks a parent to report on behavior over the past 6 months, right? Those are the kinds of things that we do. We also look at things that we couldn't expect to change in the short term. So if we had a drug for autism and I used a parent questionnaire measure that asked about friendships, that drug could be fantastic. But the kid may not have friendships at the end of the trial because those kinds of things take time, right? The behaviors that we study in autism that we wish to see change are derivative, are sequelae of the systems that we would intervene with. We would love predictive biomarkers. Again, right now we choose treatments based on, kind of clinicians' best estimates based on these clinical data that we collect, and really, truthfully, based on cottage industries. If you are closer to UCLA, you're going to get a treatment called Jasper. If you're closer to Denver, you're going to get a treatment called the Early Start Denver model. But these are not decisions that are made scientifically, certainly not based on biology. And then we would want diagnostic or screening biomarkers. And again, so we could think about this, at least in my field, people think diagnostic meaning definitional of autism. But again, reading that best framework, and this is the way that we think about the biomarker that I'll talk about today, is we could think of as being diagnostic of a subtype, of a more homogeneous subgroup of autism, which would be really advantageous. And really, I think, is where the context of use that we're pursuing and the one that is most feasible given the state of the science in autism. So what are some of the challenges? It's funny because I feel like that's all I've talked about so far. But we don't know the etiology of autism in most cases. We know a handful of single gene disorders that not perfectly, but with great likelihood, predict development of autism. So we don't know exactly what the cause is. We doubt that there is a cause. So there are probably many, many different causes of autism. Remember, we're just talking about a set of behaviors that exist in some person across development. We don't, even if we put the issue of kind of etiology, to me, connotes diagnostic bins. If we put those bins aside and just thought about mechanisms with which we might want to intervene, we still don't know a lot about mechanisms. Given the heterogeneity of autism, there could be different mechanisms implicated in different children. There could also be different mechanisms involved in the same child when we consider the diversity of functional domains impacted in autism, right? There could be mechanisms related to language, mechanisms related to face perception, mechanisms related to social motivation. I keep talking about children, but not just children have autism. So when I say autism, I could be talking about a 3-year-old, or I could be talking about a 30-year-old, or I could talk about a 60-year-old. And so what does that mean for biomarkers? Do we need, especially functional biomarkers, EEG biomarkers, can we expect us to have a biomarker that we could use across the lifespan, or do we need to be looking for different biomarker batteries for different cohorts? How do we deal with it when it intertwines with natural human development, immature brains in early life, aging brains in older life? These are things that we understand very poorly. As I said, a lot of the biomarkers so we've heard a lot about MMN, for example, at this conference. It's also a biomarker that's been pursued in autism, and it's true of many of the things in autism. They're implicated in multiple disorders. We don't know how they would be uniquely relevant to autism, or whether they are relevant to autism because they're relevant to a different diagnostic bin. As I said, we have really weak clinical measures, which is really hard. When we see, if I wanted to measure change use a biomarker to measure change in response to a treatment, how do I, without a clinical ground truth, how do I validate that that biomarker is measuring something meaningful? So that's really hard. And then lastly, for these functional biomarkers, we're dealing with kids. Often kids, they can be anybody, but by and large, we're dealing with people who may have tactile sensitivities. We're dealing with people who may be less inclined to please the experimenter or the physician examining them. And so doing an EEG, for example, on a person with autism is often different than doing it on a person of the same developmental level who doesn't have autism. It's harder. And so for example, if I do an EEG and a child is extremely distressed with the procedure, that distress is probably providing more signal in the EEG than the autism, right? And so that's another complication in autism. So where do things stand? And this will be the first time I guess I say something good. So what a great method for autism. Right, so EEG, I mean, it's a non-starter. So it's a non-starter to think about a biomarker in autism that one can't collect in people with autism, right? So EEG is great. You can use it in children. As far as brain imaging methods go, it's extremely tolerable. You have to tolerate wearing a damp hat. And it's cheap and it's accessible. Greg, who gave an awesome talk yesterday, talked about the gizmos, and I think about it almost in the opposite way. EEG, we're sticking electrodes on a person's head and we're picking up the signal, right? There's an EEG in every hospital in the country. I think there's low odds that any biomarker we'd be picking up would be specific to the system that we're using. So really, should we figure something out? It could be deployed at scale. We already use EEG at the population level for screening for seizures, for screening for newborn hearing. So I think it's a great method to pursue. I will also note, I've been giving these talks for so long, I think this is the first time I've managed to squeeze both of my daughters into one slide. So there are my EEG models. In terms of the actual, I'm not going to talk about all of them because I think there are truths that are universal for EEG biomarkers. And I think John said it in a nutshell, not really ready for prime time. But there are many markers, and I think broadly about them falling into two categories. We have EEG markers that can tell us something about a mechanism. So something like resting EEG, telling us about kind of cortical connections or broad cortical temporal dynamics. Things like the visual evoked potential, telling us something about visual perception and GABAergic activity. So those are clearly tied to a mechanism. What we don't have is any really face-valid tie to the things that are problematic in autism. They're not tied to the symptoms. And then we can flip. So then we could think about things where there's biomarkers that are really tied to some symptom domain, like the N170 that I'll talk about, which is a marker of face perception, or an N200 marking perception of biological motion with a point light display. And so these things are really face-validly mapped onto the symptomatology of autism, but we're less clear the mechanics, right? We can tie it to brain systems, but really that's about as specific as we can get. And so that's really broadly where things stand. There's many markers. For all of the markers, really, I think these are the two things that have been shown for many markers, universally reproduced for no marker, that they discriminate people with autism from people who don't have autism, none at the individual level. These are shifts in the means of groups, and that they associate with symptomatology. And again, These are the emergent truths across many biomarkers that are true for individual biomarkers, but not in every study. So that's the strong evidence, the weaker evidence, which has some, or I say suggestive because I guess I would say strong if it reproduced more consistently, but I'll say here I say limited. We don't know the specificity of the association with symptomatology. So for example, if I do a study and I show that a marker is associated with social function in autism, well, intellectual ability is going to cap social function in many of these kids. And so I'd want to show that that association is independent of intellectual ability, or we could show a dissociation, show that a marker looks intact in domains of ability in which people with autism show preserved function. We don't know that for most biomarkers. For most biomarkers, we have a very limited sense of test-retest reliability. We have a very limited sense of stability over development. So if I want to look at a biomarker as an index of, what if we have a really long trial, or we want to look at a long-term follow-up, 6 months, well, the child's brain has also changed in these 6 months, and in most ways, and for most markers, we don't know what to expect. We have no kind of frame of reference. We don't know, we don't have a good sense of how responsive they are to clinical change. And for most biomarkers, we don't know how vulnerable they are to methodological variation. I gave the example that I don't think it should matter which EEG system we use to collect a marker in autism, but I don't know that for sure. It's not known for most markers, and that's something that we would want to know and want to rule out. So here's an example biomarker, the N170. It is my favorite biomarker by far. I thought that I loved the N170 until I heard Greg talk about the MMN, and then I realized I don't know love. I haven't written about a book about the N170 yet. So this is what the N170 looks like. This is from data that we collected when I was a graduate student. It's you can't really, it washes out, but that negative dip around right before 200 milliseconds, the N170, the N meaning it's a negative going component, the 170 meaning it happens around 170 milliseconds. This was first discovered in 1996 by Shlomo Benton and Greg McCarthy. The idea is that conceptually this represents what we call the structural encoding of a human face. This is the first in a sequence of cognitive processes that one undergoes when one sees a face. This is the first one that's face sensitive. This means this is a face. Prior to this is mom's face, prior to this is a happy face, and again, we've heard about the inverse problem. It's hard to know exactly from where in the brain EEGs originate, but based on correlations with imaging studies, intracranial studies, and response patterns, we presume this reflects activity in the STS, the fusiform gyrus, and then downstream modulation of the amygdala, which we wouldn't get with EEG. This is what we see in autism. This was the first study in autism, the N170, which I did as a graduate student in 2004. So we've been looking at this for a while. We found that people with autism show a delayed N170 latency. They do have it, but it's slower than typically developing peers, which we presume to mean inefficient processing. I'm glad to talk about the theory of that. We presume it's kind of incomplete maturation of a system that requires certain inputs during development. This delay has been seen in groups as young as 3 through adults. We see that it associates with symptoms, things like social severity, things like face recognition ability, and we see that in a limited number of studies, we see that these associations are specific. If we look at areas that are not impaired in autism, that also elicit specialized brain activity and similar kinds of components, we don't see delays there. We see in very preliminary data from my lab, we see that as a person gets better in a treatment, their N170 gets faster. And then most recently from the ABCCT, we've gotten a sense of test-retest reliability, which is over 6 weeks is about 0.67 in people with autism and typically developing kids. So in November, we submitted an LOI to the biomarker qualification program based on our N170 data. The way that we framed it, so it's interesting because if you look here, right, I showed, and this is one of the things that we struggled with. It's hard, right? We love our data so much and we want our biomarker to do everything it could possibly do, but that I learned is exactly the wrong way to think about it when you're writing an LOI, right? So what we really said is, okay, what's the strongest data that we can bring to the table, and that's going to define our context of use. And it's really the way we thought about it is as a diagnostic biomarker, not of autism per se, but as a marker of a more biologically homogeneous subgroup. So maybe, for example, you can see these are the data from our interim analyses that we the data from our more recent analyses are pending, and the final data are in process, still being collected. But you can see there's overlap between the groups. It isn't that this tells us that who has autism and who doesn't. The idea is that you see this tail in autism that's virtually non-existent in typical development, and maybe those are a group of people that we could use to enrich clinical trials by reducing heterogeneity. So that's kind of the way that we're thinking about it. The kinds of things that we continue to look at in our data, really, we haven't really done a lot of exploration of our time 3 data, is understanding more about individual differences. So things like sex, things like symptomatology, things like intellectual ability, how do they influence our results? And so far, it seems to be pretty robust irrespective of intellectual ability, which is a big concern in autism. We are looking at stability, sensitivity to change, and then we really have to figure out, so if I just drew a purple box in the corner there, but where should the lower threshold of that box be? And we don't have that answer yet. So what are some of the things that should happen next? We want to see more of this. So the N170 is, so Amit talked about these beautiful and sophisticated methods, and the N170 is kind of the opposite, right? I collect 1,000 samples of data per second over 144 trials across 128 electrodes, and then I boil it down to one number per person, right? Like we're throwing away a tremendous amount of data. The good thing about it is that we really understand what that number represents. People have been using that number since 1996. In autism, there are over 40 studies looking at that number. So it's not glamorous, but it's as far as biomarkers in autism go, it is as good as we get in terms of replication. We still want to know more. The data from the ABCCT are in children 6 to 11. It would be nice to know about younger children. It would be nice to know about older children. It would be nice to know about the children that we studied in the ABCCT when they're older, to have a better sense of longitudinal development. We want to know more about intellectual disability. We capped our sample, the bottom end of our sample, at an IQ of 60, but people with autism have IQs much lower, and it would be interesting to understand how this biomarker works in that group. It would be interesting to understand more about how it works in relation to the co-occurring conditions in autism. And I'm happy to talk about it during the panel. We really an unprecedented level of methodological rigor in this study. So anytime an EEG was done, within 72 hours, our EEG experts in Seattle had watched the video and given feedback to the site PI about the quality of that data collection, right? So really tight. Every lab swapped out any pieces of equipment that weren't identical to the other labs. So identical hardware, all the paradigms were created centrally, all data analysis was central. A tremendous amount of energy and resources went into the methodological rigor, and we don't know whether that's necessary. One of the things that's cool, Diane mentioned this EU Ames project, and we actually purposefully used the exact same assay to elicit the N170 in both studies, and they were much more, I'm trying to find a different word than willy-nilly, but I can't. They were much more willy-nilly with their EEG. They had lots of sites, lots of systems, but you know what? They saw the same effect as well. And so that's actually, I think that's good. It's telling us something about the signal, but we really don't know how sloppy one could get and have the signal still emerge. As I said, we want to understand more about clinical status, and that's an inherent problem because we don't have great clinical ground truths. We want to understand more about stability in the longer term, right? So I said the ICC over 6 weeks, we have data for 6 months, and then sensitivity to change. It's also one of the things that we're struggling with is what does that mean? So I'm not talking about eye tracking biomarker today, but we have an eye tracking biomarker that we looked at. It's incredibly stable. It's like 0.8 over 6 months. Is that a good thing? I mean, do we want to use a person's, eye color, for example, as a predictor of treatment response? Probably not. So if children are changing over 6 months and our biomarkers aren't changing, we don't know what that means. Conversely, if a biomarker is moving up and down, is that capturing something that our behavioral measures are missing, or is it not? And then, in line with what I said before, really, I want to send my data to Dr. Etkin and have him look at it, but that would be the first step, right? Is that we start to establish some predictions for some of these more sophisticated methods so that future studies like this, the ABCCT, can look at it with the same level of rigor. So that's the stuff I want to acknowledge the group. It's a broad group. This is just the leadership. And because of some convention that is probably flawed and I don't really understand, it only shows the scientists in the consortium, not the scientists at FNIH and NIH who have also been integral, but Linda and Bill and Lisa, Alice, and Meg have all been really involved, and Rosa have all been really involved in carrying out this work as well. It's a cooperative agreement, so it's really been a team effort, and I'll stop there.
Thank you. So I want to start by welcoming Jay Gupta to our panel discussion. So I just want to briefly introduce him. He has a master's in biomedical engineering at the FDA. He is serving as the chief of the neurodiagnostic and neurosurgical devices branch within CDRH. And so welcome. Thank you very much for joining us as panelists. So I think it would be very nice to have people give their high-level thoughts about what they heard about the EEG biomarkers and its potential here. And I don't know which end of the table we would like to start on, so I don't want to put anyone on the spot, but would you like to start, Jay? Thank you.
Sure, I can start. So I think, you know, a number of the things that were brought up in these EEG talks mirrored a lot of what was discussed previously. And so I don't know that necessarily there are a lot of unique aspects to EEG. You know, maybe the way that we consider certain questions and consider certain problems is unique to the actual, you know, platform of how we're collecting the data, how we're analyzing the data. But really, you know, some themes have emerged in my mind, and I wanted to just sort of touch on a couple of them in order to stress some points. So one was about variability in the data. One was about, you know, variability in EEG data or whatever biomarker data you are collecting, you know, questions about sample handling, preparation for EEG. It's, you know, questions about, are there differences between devices? Theoretically, hopefully, if you're seeing a general, you know, P300 signal or an N170 signal, that signal is going to be relatively robust. But do all of the different systems, if they're used, you know, in various different ways, you know, how well does that signal appear? And does it appear in a way that is as clinically meaningful for the person who's interpreting the data or your automated algorithm that's taking a look at that data? Those are questions to consider and think about, and I think that's been brought up in a fair amount of detail throughout these 2 days. Again, not unique to EEG, but proper validation, verification, primarily clinical validation on an independent data set. You know, a lot of the methods that we look at in order to try to determine a potential biomarker is taking a look at, you know, cross-validation. It's taking a look at potentially in, you know, a large, more general patient population. It's trying to figure out, okay, what are, you know, half a dozen or a dozen potential biomarkers, and then, you know, it's important to then look in a specific patient population and independent data set to evaluate each one of those particular biomarkers. Again, not unique to EEG, but very problematic or challenging in the neurospace, and specifically for neuropsych, is the determination of the clinical reference standard, and sort of understanding exactly what patient population we are looking at and what we're evaluating the performance of that particular biomarker in. So those are some key things. I also noted a couple of things that Dan Kranak actually mentioned earlier that I liked. One was about the balance of using cutting edge technology. So I know, you know, mentioned here a couple of talks about sort of these high density nets and things like that. Again, this ties back to the technology issue of, you know, yes, there are advancements. Yes, you know, if you're measuring something from a particular spot on the scalp, ideally that's all the same. You know, we need to actually take a look and make sure that that actually translates and is generalizable, and also that, you know, if there are particular potential human factors issues between different, you know, systems, are you actually getting a signal where you think you're getting a signal? Is it coming from somewhere else? Those are some questions to ask. I think for now I'll leave it at that.
Well, thank you for this opportunity to provide some high-level thoughts on the high-level thoughts that I just provided. I'll say some things that are unrelated. So, I talked about co-occurring conditions as a problem. I don't think that they're really a problem. For example, Alan had to leave, but Alan and I have collected data applying some of the same biomarkers in autism, schizophrenia. I'd love to do the same with Ty, and I think that's just fine, right? I think that when we think about the kinds of symptom domains that are impacted and the kinds of treatments that we'd wish to apply, they're not disorder specific, right? So if there's a way that we can predict a response to depression in depression, it's very interesting to think about how we might apply that in depression in autism. And so I think that, looking at these data in a cross-diagnostic way is useful. And then I'll make just kind of a practical comment. So I am a clinician, I run a clinic, and we're here thinking about biomarkers, and the first applications are going to be in trials. But, the practical implications of the technical difficulties I described in autism have really, really significant public health implications, right? So that right now, to get a diagnosis of autism, the rate limiting factor is the availability of expert clinicians. And this play session that I talk about, the play session, it costs, it's a box of toys that costs $1,000, right? And so the idea that we could figure out ways that could be deployed in a hospital setting with a few electrodes could really, down the line, have really dramatic public health implications, not just the kind of academic implications that I think that I talked about earlier. So I'll stop there.
Well, I want to also thank people for the invitations to come. Bill, others, thank you. It certainly was very informative to hear different perspectives of the challenges of biomarkers from people in the FDA and people in the industry and people in academia. And I do think it's really important to pull these people together to talk. So it's a good thing that's happening here, and I'm glad to be part of it. I think the thing that strikes me very much about the data that you heard from the three of us is that for the first time in my life, and I've got enough gray hair for that to be a long time, I sat on a series of talks where all of the people were not presenting group level data and sitting down. Everyone was trying to have large sample data to explore heterogeneity and to find ways to use that. And that's real. That's an evolution, and I think it really reflects the desire of many of us working in this area to try to bend our knowledge into ways that are more clinically useful. And I think that's really exciting. I think the studies with autism, trying a subgroup of people distinctive on their processing of face information is really very interesting. I think our separation in BSNP of high excitability and low excitability pyramidal cell activity in subgroups of patients that do not differ much in their cognitive profile and do not differ at all in their symptom profile is really very exciting. We're finding our syndromes and parsing biologically discrete subgroups of people and processes that are much more heading towards something to be targetable. And I just think that's really exciting. And I think as we partner with industry and find ways to implement these tools, and we continue to identify the molecular mechanisms that are associated with the different subgroups that we have, there's really exciting promise for our field to make significant advance in drug development, which for those of you working in neuropsychiatry has been pretty much stalled for a long time. And I think this may give us a really big leg up to be able to allow us to have much more successful trials in this area. So thanks again for the invitation.
So this is actually, I've been excited about EEG in the past few years as we've transitioned the lab from fMRI heavy to EEG heavy. But seeing these talks and Greg's yesterday, actually, I'm even more excited now because there's a lot there. And I think, my kind of higher level view is that there's actually a lot of data out there. And one can't really say that for a lot of modalities. And that's an opportunity. So that's an opportunity around data sharing, around thinking about harmonization. What kind of features, back to what Jay was saying, right? What kind of features make for a good data set? What kind of ways could you automatically rule out a data set so you can have real criteria? And then how do you pool across data that are collected in diverse places, be they neurology clinics or psychiatry departments, research studies, clinical care. There's probably, I don't know, I'm guessing 500,000 to 1 million EEG data sets out there easily, right? So can they all just come together and hang out in the same database? It seems like an opportunity, and that requires an organizing body, I think, to make that happen.
So this was great. Thank you very much. One of the things that came to mind after watching Jamie's talk and hearing what Jay had to say, what do you think about the need for a normative database? When we look at something like blood pressure or LFTs, we have a kind of an idea of what normal values are across ages. And do we need something similar for EEG or any biomarker in order to start qualifying things? Just like to know what your thoughts are about that.
So I can imagine the neurodevelopmental folks will take a very different view. I actually don't think that one is necessary for certain applications. So in our prediction of outcome, let's say you get a biomarker predicting outcome in whatever, we have rehab for addiction, antidepressant treatment with meds or RTMS, psychotherapy for PTSD. In all of those cases, we look at healthy people and patients on that signal, and they don't differentiate. So I think it's actually more to be driven by the context of use. So in our context of use, it just turns out that that this feature of the brain that predicts outcome doesn't vary by disorder. But I can imagine actually for other indications that may be completely different logic.
Yeah, I agree. I think, for example, for the purpose, the context of use that I described, it doesn't really matter, right? It's relative to other people with autism, the difference. If we thought about a context of use where this could be telling us something about risk for the disorder, then normative databases would be really helpful. They'd be really complicated, right? Because for the kinds of things that, at least that we're studying, I presume things like variation in IQ, variation in language ability, variation in anxiety level are going to move the biomarkers around, so it'd be hard to know. But I guess it's true for every normative database. Those things exist in there, and you have enough that there's kind of an emergent truth, but it would have to be large.
I think that while we have many disadvantages working in neuropsychiatry, one of the advantages we have is that the age of onset tends to be around 20, and we've mostly studied people up to the age around 60. And in that age range, the developmental age-related changes on most of the things we look at in MR and EEG are relatively modest. And I think as long as we're doing consortium studies as we are now, that include hundreds of control people where we can see that where the range of normal is and how far outside of that. I think we're in a good place to start for trial application. I suppose if you got into clinical application where you want the mean and SEM of what you'd expect a patient to be on a given laboratory value for medical decision making, then if we want to get to that point, having a lot more data about the range of normality could be more informative for that purpose.
So maybe kind of branching off that a little bit, also maybe want to take, this is my opinion, feel free to disagree, and this may not come as good news to pharma, but at least my sense is because we're so poor in defining our disease entities in psychiatry, we're probably not going to see any miracle drugs anytime soon in the standard one-size-fits-all approach. So in some ways, there's actually advantages for all involved to just say, you know what, let's get rid of those diagnostic labels. They're actually, as you saw across all of these talks, not helpful in describing biology, not helpful in predicting outcome. Maybe the place to start is actually a biomarker, because autism, depression, schizophrenia, a clinician will know that this person has a problem needing treatment. The question is, what is it in that person that you can connect to a treatment? And maybe we should just take off the shelf all of our DSM misdirection over these years. This is like hopefully not recorded, right? No. And anchor it in something that's measurable and objective, and have criteria as a group to move forward on that front. I'd actually be curious what FDA says. It might be a more reliable measure, right, than a diagnosis in some ways too.
So the one comment I will make is that, one of the, it's not necessarily unique to collecting or creating a normative database, but when you're now dealing with these, like understanding these types of measures that may not be, there may not be a clear relationship to the clinical ground truth as defined by the behaviors or as defined by DSM or things like that. I mean, you're going to have a very robust understanding of the signal that you are measuring and the, whatever biomarker you're defining. So in that sense, some development of normative data, of understanding what is responsible for variation, understanding, why this signal is important, those types of things are going to be of increased importance in that sort of case.
I think that was a really important point, and I think that there's a growing tendency towards heresy amongst people doing biomarker development with regard to our progressively reducing respect for the DSM diagnosis classifications in terms of how they are going to advance our ability, how they're going to limit and impact our ability to develop better treatments in the future. So treatments that are aiming to treat all depressed patients or all schizophrenia patients, it's just a problem. It's too big a target. It's too heterogeneous. There's no biological process associated with these syndromes that is targetable by a drug. So narrowing things down into smaller groups, which has really been the avenue to success in most fields of medicine, is something that we really need to do. And we don't have fluid biomarkers. The genetics aren't paying out. And it's really these EEG and MR biomarkers that are giving us the leverage to do that. And that's, I think, the place for us to start.
Okay, Tiffany Farchione.
So, hi, Tiffany Farchione. I'm the acting director of the Division of Psychiatry Products at FDA. And given that I saw both Linda and Bill look at me when you made the comment about, you'd be interested to hear what FDA had to say, I figured I should probably come up to the mic. But, when it comes to the DSM, I mean, everybody can agree there's nothing biologically meaningful about the clusters of symptoms that are described in the DSM. I would actually look forward to the day when maybe we have to rewrite our labels based on biology. We're not there yet, but the closer we can get to having some sort of biologically meaningful, druggable target, the better off we all are as a field. So, it's not that provocative. It's something that I would look forward to. So, thanks.
I just want to add, as a clinician, I think the DSM has utility. I think that maybe the trick is to change what it is that we think that we're treating, right? So that we recognize that saying autism or saying schizophrenia is a meaningful thing for us to communicate about a patient, for example. But to say you're going to treat autism is totally meaningless, right? You could say that I'm going to treat social cognition, and it doesn't matter whether the person has autism or schizophrenia. But I think that there's also kind of, I agree, we're not ready to get rid of it yet, but it's obviously not.
And we see that in other areas of medicine, right? I mean, we don't use catch-all terms, except maybe in how we communicate to patients as physicians, right? When we talk about heart failure or cancer, like, you have cancer. What does that mean? It doesn't inform my treatment at all, right? But it's exactly meaningful for a patient. So I'm not saying get rid of how we communicate, like you have a N170 deficit, congratulations. But it's understanding which party needs what, right? And what we need is actually going to be different than how we communicate that to people.
Joe, you're next.
Yeah, Joe Manetsky, FNIH. So we're talking about. I'm thinking back to the depression slides of the within the first discussion, and the reanalysis to get an opposite response. And I think the thing that hit me the most was not the 30% change, between treated and placebo and not placebo. It was the 40 or 60% change of the placebo arm itself, or it was 40 probably. And that's kind of the death knell for most, pharmaceutical projects, because now you're looking at a huge amount of annoyance of all these people who get better just because you tell them to get better. So I'm wondering if any of your analyses actually addressed that issue of, are there differences within that placebo arm, those who seem to show an effect and those that don't? And that could be for any disease, psychiatric disease.
Yeah, so I didn't show that, yes, we actually can develop predictors for the placebo arm itself that differ from sertraline, so you could stratify on that. That would be a little odd in some ways if you think about the label implications, because you're not actually studying the drug but reducing the placebo response. That has to be creatively thought about, I think. And we know by pooling the sertraline and the placebo arm that there's not that much of a common prediction across the two, so they really do seem different. I'm not sure that, placebo rates have definitely been creeping up in depression, and that's a problem. I'm not sure if solving that is necessarily the real issue, as much as what has been kind of mentioned, that the biological subgroups are distinct, that it should be very easy to separate if you can get the right biological subgroup, even with a high placebo response rate. So I think they're two kind of different things, but it certainly seems from our data that placebo and drug are not doing the same thing biologically, not predicted by the same thing biologically.
As someone who's been living in the intersection of academia and bringing EEG into multi-site clinical trials, I really loved your presentations, all of you, and the people this morning as well, and yesterday, of course, too. I wanted to highlight a couple things that came out in the presentations today. One, I mean, in your presentation, you showed that spatiotemporal relationships matter and that they're very important. In the morning before the session start, Bill asked me a question that I think was posed to him by Ty, which is, if you're only going to show data from one channel, why bother with collecting data from 64 or 128? And the answer is really simple. It's the spatiotemporal relationships. That is, that when you have data from a lot of sites, you can very purely and mathematically cleanly remove sources of error, sources of noise and artifact. And in doing that, you can substantially improve your signal-to-noise ratios, your reliabilities, your sensitivity to detect drug effects. Related to that, Jamie, you talked about like the details and how the details matter, how the equipment matters, the oversight matters, the QA process matters. And in our kind of industry-sponsored studies conducted, in lots of different places, anything that you can do to reduce the error on the other side will increase your signal-to-noise ratios. And so, like working with Neurosig, and where they have like dedicated engineering, they have software, they have 24-hour tech support, they have QA, they have processes in place, they have like basically immediate feedback. It makes a difference at reducing variability. You also mentioned, how much I love MMN. And it's true, I do think it's a great measure. I think it's a great measure. I like lots of other measures too, but the one thing that I really like about it is that it has very high test-retest reliability. So we use lots of different things, but with ICCs of 0.9, you can do stuff with that for looking at change. And so if you can reduce the number of subjects required for an Alzheimer clinical drug trial with an experimental therapeutic, you can save a couple $1 million on the study budget. You can reduce the number of sites required. If the ICCs are in the 0.5, 0.6 range, that's a non-starter. So like if somebody in the room came and said, we want to do a study of, new measure X that I saw at a poster at ACNP, I would say, what is the reliability? And if it's not up there, I can't recommend that we do it. And so Anything that you can do to increase those ICCs. Bill also asked, like, what is the incremental value of improving the ROCs, right, in the morning from 0.73 to 0.78? Well, an area to focus on is improving the ICCs. If you can improve those ICCs first, you can probably improve the ROCs or AUCs or whatever kind of curve types of metrics you want to use. But, you know, I think we're getting there. I like the idea of not relying on DSM categories, but to really use direct and reliable assays of brain function to identify subgroups of patients, like John beautifully demonstrated, and going forward from there. Just one last point about, like, there's EEG systems in every hospital. It's true, and a P300, you know, in one system should be like roughly the same as the P3, but the systems, the analyses, all of those have different psychometric properties, and again, those psychometric properties really matter. So thanks to everybody here, because it's important work, and I think we're getting closer to using, like, the tools of neuroscience to better understand brain disorders and better treat them too.
Yeah, thanks for great sessions, and I learned a lot over these 1.5 days. So my name is Stefan Pont, and I'm with Boehringer Ingelheim. So we do actually do EEGs in our studies and a number of other stuff, and I'll speak about some of that this afternoon. But one of the things that always makes me extremely nervous when we need to decide whether we want to include this in our studies is that when you start looking in the details of the protocols on how to do it, it's all over the place. You mentioned mismatch negativity, and it's a great biomarker, and actually Greg sold us on it, and we've decided to pursue that in all of our programs. But then what sort of deviance? And then it's double deviant, and you show triple deviance. So what is it? And I think one of the lessons I learned when I started in industry and did electroretinograms for a certain reason was that a group of scientists that were involved in that, academics, came together and said, this is the core protocol. Do whatever you want to do at academic level beyond it, but at least this is the standard core protocol of how to do it. And it would really help us in the field of drug development if people that are experts in this area could come to a sort of core protocol. Is it 64 leads or 32 or 23? What would you recommend should be the standard? Because then we can do it across all companies and give you the placebo data. I'm sorry? No, but I mean, I think he has an industry background, so I understand the pain. I mean, because you pointed out that it's cheap, EEG is not cheap. Okay, EEG is cheap if you do it in the clinic, but if you do it in the context of a study, there's issues of data integrity. You mentioned data quality. That costs a lot of money, and it's actually as expensive as fMRI to some degree. So my plea is please think and consider working out a standardized protocol, and we can start from there.
Thank you. That's a really, really good point, and I think that that's what we've heard as a lesson learned from all of the other types of biomarker technology platforms we've heard about earlier in the week. We're talking about proteomics, we're talking about PET imaging, we're talking about all sorts of measures, functional MRI, connectivity, what we really need, and that's what I've heard. That's like an overarching comment for the entire biomarkers meeting that we've had is that using a standardized protocol, taking into account that at some point you have to call the technology good enough to standardize and have it be acquired and collected and shared with similar processing pipelines so that it can be shared and mapped across different diseases, and having the opportunity to learn from how a measure is working in Alzheimer's disease versus, neurodegenerative disease versus psychiatric diseases. I think there's a lot can be learned, and I think saying, that it seems like we're at a critical junction now where that would be really important, because some of these things look like they're ready to be deployed in the industry setting in clinical trials, and trying to learn about how they perform and whether they can be mechanistic or whether they're more, identifying substrata for enrollment in studies and for directing to specific mechanisms. It seems like the time has come, and that seems like a really important consortia effort that could be undertaken in many different levels. And so I'd like to hear what others think about that idea.
I think I agree, very much agree with that. And I think it's a really good point that was raised. I think that standardization has a lot of appeals, and I think clearly there needs to be discussion for this. But I will point out that there are some limitations. You would not want to standardize a battery of 3 blood tests for all the studies that you want to do in neuroscience, and so different paradigms are going to be sensitive to different processes that you might be targeted by a drug that you would know a priori. So the idea of having a standard toolbox that would be used in all drug company studies across psychiatry seems not likely to pan out in the long run in terms of demonstrated utility for a context of use. I mean, I think but with that said, there are simpler, straightforward things that we would, I think, all pretty much agree to have a resting state EEG study, to have a simple tone response, to have a simple mismatch study. To do MR studies with a resting EPI study, a T1 study, a DTI study. There's a list of things that I think people would agree to pretty quickly that should be included. Now, I think if you when you get down to the point of doing a trial, a significant phase 3 trial, it was probably not a bad idea to talk to a few experts in the area to get input about, given the particular target and the particular knowledge about a particular drug and the particular set of preclinical data that you have available, what might be what might supplement the standard group of things. And I think as we get going with this will evolve. But I think we are at a point where we could take that step. And if you'd have a meeting and you'd probably have some food fights about the details amongst the people, but I think the consensus would come pretty quick at this point.
Hi, Dan Kranek. I just want to add on to that. And Dan Barboriak, please feel free to add in. The brain tumor group, I think, did this in a good way. It's not saying what elements you necessarily to have in all trials. It says, if you collect this type of data, please, please, please do it this way so that we can all learn from it in the future. And also the advantage there is to kind of have this idea of a basic and an advanced protocol. So it says, please, please, please do it this way for the basic thing. If you want to add on other stuff, great. That's fine. That allows for learning more, but it's not to specify which elements are included. It's to specify if you collect this type of data, please collect it in a standardized format. So I just want to emphasize that.
I think we don't want to get hamstrung with a fixed thing that we would all try to do, and industry says this is the thing we're going to do in our studies in the neuroscience space. I mean, I think that is a problem, but I think having a standard subgroup of things that would be relatively routinely included in a relatively standard way, ideally quite standard way, it would be really important. So I just emphasize and agree and say, boy, that was a good thing you just said.
So thanks. This has been a great discussion, and I just wanted to mention how I'm excited to hear what Tiffany's thoughts are about maybe changing labels, because when it comes down to it, if we start to define subgroups in certain ways, we still have to write a label. So the idea that the FDA is open, I'm open to it, Tiffany's open to it, you've heard it here, that we're open to discussing different ways of describing diseases rather than just DSM-5. Because I think we would get into a vicious cycle if we're waiting for DSM-5 to change so we can write the label, but drug companies aren't willing to do the studies because they have to write a label that goes with DSM-5. Nothing really gets done. So that was my parting thought.
Well, thank you very much. It's been a wonderful discussion. I think now we're going to go ahead and move on to the lunch and come back at 1:15. Or do you want a little 1:25? Okay, so we have now until 1:25 for lunch. Thank you, Mike. Sorry.